on the temperature used for sterilization, moist heat sterilization is categorized into three: temperature below 100 degrees Celsius, temperature at 100 degrees Celsius, temperature above 100 degrees Celsius. Okay. So, first, temperature below 100 degrees Celsius, in which the most commonly used technique is pasteurization. This technique is mainly employed for the sterilization of milk. The milk is heated at either 63 degrees Celsius for 30 minutes. This method is known as Holder method, or otherwise the milk is heated for 50 to 20 seconds at 72 degrees Celsius. This process is known as flash process, followed by quick cooling. Actually, by this process, not all the microorganisms are removed from the milk. The milk may contain both beneficial as well as pathogenic or contaminating microorganism in it. This process removes non-sporing pathogens such as Mycobacteria, Brucella and Salmonella. But pathogen like Coxiella burnetii, which is relatively heat resistant, so which may survive this Holder method. That is one of the defective of using pasteurization technique for milk. Vaccines of non-sporing bacteria, they can also be sterilized by using a temperature below 100 degrees Celsius. So here a 60 degrees Celsius for 1 hour is used. Okay. Practically all mesophilic means medium temperature loving microorganism. So all mesophilic non-sporing bacteria, they are killed by exposure to moist heat at 60 degrees Celsius for 30 minutes. Okay, but bacteria like Staphylococcus aureus, Streptococcus faecalis, which require 60 minutes at 60 degrees Celsius. A temperature of 80 degrees Celsius for 5 to 10 minutes is required to destroy the vegetative form of all bacteria, yeast and molds. Among the most heat-resistant bacteria, the spores of Clostridium botulinum, which is highly resistant, which require nearly 120 degrees Celsius for 1 hour, 120 degrees Celsius for 4 minutes, or 100 degrees Celsius for 330 minutes to destroy them. Most of the viruses are inactivated very rapidly at 60 degrees Celsius, but some are relatively resistant. For example, poliovirus, hepatitis B virus, which may survive 30 minutes and over 10 hours respectively at 60 degrees Celsius temperature. Next is temperature at 100 degrees Celsius, in which first technique is boiling. It is a routine practice we are following in our kitchen. So vegetative bacteria are killed almost immediately when we are boiling the water or boiling the sample at 90 to 100 degrees Celsius. But sporing bacteria, endospore producing bacteria, require prolonged period of boiling, and this boiling technique is not at all recommended for sterilizing certain instrument, especially used for surgery. So for the effective boiling, the materials to be sterilized should be immersed in the water, then it should be boiled for at least 10 to 30 minutes. And the lid of the boiler or sterilizer, it should not be kept opened during the boiling period. Second technique is steam at atmospheric pressure. That again 100 degrees Celsius is used, but the technique is steam at atmospheric pressure. And an atmosphere of free steam is used to sterilize culture media, and here a Koch or Arnold steamer is used for the purpose. Okay, laboratory autoclave can also be used here. It is an inexpensive method, and here the container and medium is simultaneously sterilized. Structurally, this steamer consists of a tinned copper cabinet. Within the cabinet, proper level of water should be maintained. Over the water level, we should keep a perforated tray in which we can keep our properly wrapped sterilizing materials, and lid should be properly closed during the sterilization process, and the lid is conical in shape. Okay, a single exposure of 90 minutes usually ensures sterilization, but for media, especially containing sugars or gelatin, an exposure of 100 degrees Celsius for 20 minutes on 3 successive days is used. This process is particularly known as Tyndalization or intermittent sterilization. Why 3 successive days sterilization is required means there is a basic principle behind that. When we are first exposing the materials to steam or during the first sterilization, it kills all vegetative form of bacteria and spores, and since they are in a favorable medium, there is a chance to germinate this if any spores remained in the medium, so they were further killed on subsequent sterilization. But still some spores of anaerobes and thermophilic bacteria may survive this process. Next technique is steam under pressure. Here more pressure along with the temperature is used for sterilization. Don't be mistaken, this image is given here related to Arnold or Koch steamer. Okay, and steam under pressure means this technique basically here higher temperature and pressure is used and it ensure proper sterilization. The major instrument used for steam under pressure sterilization is autoclave, and in which usually a temperature higher than 100 degrees Celsius is used. The basic principle of autoclave is that when a pressure inside a closed vessel increases, then the temperature at which water boils also increases. Both the temperature and pressure effect together helps for the proper or highly efficient sterilization. Usually while autoclaving, moist steam is used for sterilization, right? So this steam when it touches the cooler surfaces over the materials that we kept for sterilization, this hot steam condenses to water and its heat is transferred to the material. So in such that way it sterilizes our material to be sterilized. Sterilization by steam under pressure or autoclaving usually carried out at temperature between 108 degrees Celsius and 147 degrees Celsius. Usually aqueous solutions are sterilized between this 108 degrees Celsius and 120 degree Celsius. Okay, and several types of steam under pressure sterilizers are there, but laboratory autoclave is the most commonly used technique in microbiology and other laboratories. So simplest form of laboratory autoclave, either it is vertical or horizontal type cylinder. It is made up of gunmetal or stainless steel and placed inside a iron case, and its lid or door is tightened or fastened by means of screw clamps, and it is made airtight. Then the autoclave on its lid, it contains certain other structures like discharge tap, pressure gauge, safety valve, etc. So this autoclave is heated by means of gas or electricity. Next, how we can sterilize things by using autoclave. So first of all, sufficient water should be added to the autoclave cylinder. Then materials to be sterilized is placed on a tray. Then the tray should kept inside the cylinder and close the lid. Then autoclave is heated. Okay, and the lid is screwed tight and keep the discharge tap over the lid open. Okay, then the safety valve, I told that there is a safety valve on the lid. Though this safety valve is adjusted to the required pressure, usually 15 lb pressure is used. It is already set in the autoclave. Initially, while heating the autoclave, what happens means, I already told that we should keep this discharge tap open. So through that discharge tap, steam air mixture from the cylinder is allowed to escape freely into the air. So until the complete steam air mixture is removed from the cylinder, we should wait. Thereafter, we should close this discharge tap. Then slowly this steam pressure and temperature inside the hot air oven rises, water boils, and until it reaches the desired set level, usually 121 degrees Celsius and 15 lb pressure. Then it is set in the autoclave, so that the when reached the desired level of pressure and temperature, automatically the safety valve opens and the excess steam escape. From this point, we should calculate the holding period. Okay, usually 20 minutes is used as a holding period. When the holding period is over, the heater is turned off and the autoclave is allowed to cool. Here you can see a marked diagram of a vertical autoclave. One shows the release valve, second shows safety valve, and third shows the pressure gauge. It indicates the pressure inside the vessel. Fourth is the screw used to tighten the lid. Fifth is the cover. And sixth one is the body. That means iron case within the vessel is present. Seventh is our tray which containing the materials to be sterilized. And eighth shows the heating element. And ninth is the stand to place the autoclave properly on the floor. There are some disadvantages for this type of autoclave. First one is that in this method, air discharge is inefficient. If the complete air inside the vessel is not initially removed, then the desired temperature will not be attained. Second is that in this autoclave, there is no facility for drying after sterilization. So it takes little bit time for drying our materials. Next is sterilization control. Similar to our hot air oven, here in autoclave also certain sterilization control measures are followed in order to determine the efficiency of sterilization. First, biological method. Here spores of Bacillus thermophilus is used. The spores were impregnated in a strip and kept along with the materials to be sterilized inside the oven. Inside the autoclave, and its spores require an exposure of 12 minutes at 120 degrees Celsius to be killed. So after sterilization, the paper strip impregnated with the spores of this bacteria should be removed, and it should be inoculated into proper medium, and whether any colonies are formed, that we have to check. Colonies are produced, that means the sterilization is not proper, not the desired temperature is reached, and that's why the spores were germinated. And if the spores were not germinated, that means the desired temperature is attained and the sterilization is proper. Along with the biological method, some chemical indicators, autoclave tapes, and thermocouples may also be used for checking the efficiency of autoclave.